Bismillahirrahmanirrahim. Today we are going to talk about physiology of the menstrual cycle. I'm Dr. Manal Hamdy, Professor of Obstetrics and Gynecology, Cairo. The normal menstrual cycle, this is a tightly coordinated cycle. This is of a stimulatory and inhibitory effect, result in the release of a single mature oocyte from a pool of hundreds or thousands of oocytes. And there is variety of factors contributing to the regulation of the process. This is including hormones, paracrine and autocrine factor. Here it is. And the average menstrual cycle lasts for 3 to 7 days, mean 5 days, recurs every 21 to 35 days, mean of 28 days, and it has an average blood loss of about 30 to 80 ml per cycle, with the first few cycles following menarche and those prior to menopause, usually irregular, of course mostly ovulation. And here are the characteristics of the normal menstrual cycle. Normal every 21 to 38 or 21 to 35, should be regular with normal duration of flow and normal volume of monthly blood loss. We have other descriptive terms. Maybe it's infrequent, maybe it's irregular, short cycle, or light volume. Along the lifetime from puberty to menopause, women will have around 400 menstrual cycles, and each cycle represents a complex interaction between hypothalamus, pituitary gland, ovaries, and endometrium. With the hypothalamus, this is a small neural structure situated at the base of the brain, above the optic chiasm and below the third ventricle, and it's connected to the pituitary. The hypothalamus release what's called hypothalamic releasing hormones. They are small peptides. They have short half-life of few minutes due to rapid degradation. They are released in minute quantities in pulsatile manner. They reach the anterior pituitary via the hypothalamic portal circulation or the hypothalamic hypophyseal portal system. And the biologically active concentrations of these factors, they are locally restricted to the anterior pituitary with very low concentration in the peripheral circulation, so they are undetectable in serum. So how do I know their presence and activity? The level of the corresponding pituitary hormone, they act as marker for their presence. And the hypothalamic releasing hormone include the gonadotrophin releasing hormone. This is a decapeptide. This is formed of 10 amino acids produced by the arcuate nucleus in the hypothalamus. Here it is, the arcuate nucleus, and is responsible for two things: the synthesis and release of pituitary gonadotrophin, the FSH and LH. Then we have what's called the thyrotropin releasing factor. This regulates the secretion of TSH. The corticotropin releasing hormone, this stimulates the release of ACTH. We have the growth hormone releasing hormone. This stimulates the release of growth hormone. And the prolactin inhibiting factor, it inhibits the production of prolactin, so oral stimulatory, except the PIF or the prolactin inhibiting factor. And the pituitary gland, this lies at the base of the brain. This is the pituitary gland below the hypothalamus. It's situated within a bony cavity called the sella turcica, and it has two major portions. We have the anterior pituitary and the posterior pituitary. With the anterior pituitary, this is the anterior lobe, this is the intermediate, and this is what's called the pars tubalis. The posterior, this is the pars nervosa and the infundibular stalk. The anterior pituitary, or what's called the adenohypophysis, it consists, as I told you, of anterior lobe. This is what's called the pars distalis, intermediate lobe, and the pars tubalis around the neural stalk. It's derived from the ectoderm directly connected to the hypothalamus through the hypophyseal pituitary portal system, and it represents a major route of the transport of the hypothalamic releasing factor we talked about a few minutes back to the anterior pituitary gland. Then the anterior pituitary primarily serves in the production of 6 hormones produced by 5 cells. FSH and LH, this is produced by the gonadotropes. With the FSH and LH, they are glycoprotein. The responsibility is the gross maturation rupture of the Graafian follicle and maintenance of corpus luteum function. TSH, or the thyrotropin stimulating hormone, produced by cell known as then ACTH, adrenocorticotrophic hormone. This is produced by adrenocorticotropes. Growth hormone by the somatotropes. Prolactin produced by the lactotropes. As I told you, pituitary hormone are all stimulated and controlled by the hypothalamic releasing hormone, exception of the prolactin under chronic inhibition by the PIF, or the prolactin inhibiting factor. So any damage to the pituitary stalk will result in hypopituitarism, with the exception of increase of prolactin. Posterior pituitary gland, or the neurohypophysis, from the name it's derived from neural tissue, directly continuous with the hypothalamus and the CNS, and it consists of the posterior lobe, or the pars nervosa, and the neural stalk, or the infundibulum, and it serves primarily in the transport of oxytocin and the vasopressin, or the ADH, the antidiuretic hormone, along the neuronal axons from the supraoptic and paraventricular nuclei to the release into the cell. The ovarian cycle, the fetal ovary contain maximum number of 7 million primordial follicles at mid-gestation. The number declines sharply, reaching about 1.5 to 2 million at birth, then decline gradually to reach 400 primordial follicles. And throughout the menstrual cycle, the ovarian follicle become gradually exhausted, become depleted by the time of the menopause, and the ovarian cycle is divided into follicular phase, luteal phase, separated by the ovulation. This is the ovarian cycle. We will talk about it from the primordial follicle to reaching ovulation and formation of the corpus luteum. We have the changes in the anterior pituitary hormone and the ovarian hormone. Then the ovarian cycle, this is the menstruation, this is the build-up, and this is the alteration of what's called the proliferative phase and the secretory. What's the two-cell system? What? The two-cell, TWO-GONADOTROPHIN hypothesis of estrogen production. We have within the ceca cell, this is the ceca cell, the LH stimulates the conversion of cholesterol into androgen. This is androstenedione and testosterone, and this androgen, they are transported from the ceca cell to the granulosa cell. What happened in the granulosa cell? FSH stimulates aromatization of these androgens, to estrogen by the enzyme aromatase. They form estrone and estradiol. The estradiol, this is the chief estradiol, this is the chief estrogen, ovarian estrogen. What's the function of this estradiol? It enhances the induction of FSH receptor on the granulosa cell, and the estradiol acts synergistically with the FSH to increase the LH receptors on the granulosa cells. The androgen produced within the ceca cells If they are produced at low level, they enhance the aromatization and hence increase estrogen production. If they are produced at high level, they inhibit the aromatization and produce follicular. What's the control of steroidogenesis? Apart from the FSH, LH, estrogen, and androgen, sorry, some autocrine and paracrine mediators play important role in the process. We have inhibin, this different type, inhibin A and B, activin A, B, C, AC, and E. Inhibin, what's the function of the inhibin? Inhibin, this is a peptide produced by the granulosa cell, and it attenuates FSH production and enhance the LH-induced androgen synthesis. So the dominant follicle continue to grow or to develop, and other follicle undergo atresia. Activin, this is the opposite action of inhibin. Again, it's a peptide produced by the granulosa cell in the pituitary gland. It augments the FSH production, so the action is opposite to that of inhibin. We have growth factors like insulin-like growth factors, fibroblast growth factor, epidermal growth factor. They all enhance the responsiveness to F. How is the dominant follicle selected? During the folliculogenesis, a dominant follicle is selected. This is continue to grow, mature, and ovulate. Other will be arrested and later become atretic follicle. The selected follicle is characterized by they have the highest number of FSH receptor in the granulosa cell. They have the most efficient aromatase activity, and they have the highest concentration of FSH-induced LH receptor and the greatest amount of estradiol and inhibin produced. Thus, they require the least FSH and LH level to complete maturity. This is the approximate concentration of pituitary and ovarian hormones during the normal menstrual cycle. Pre-ovulatory follicle, mature Graafian follicle. The oocyte is pushed to one side of the follicle, secretes gelatinous material around itself, forming the zona pellucida. Simultaneously, the ceca cell become differentiated into ceca interna and ceca externa around the multiple layered granuloma. This is ovulation. Late in the follicular phase, FSH results in the production of increasing amount of estradiol by the dominant follicle. This will induce the LH receptors on the granulosa cells. This high estradiol level, it exerts a positive feedback on the pituitary gland with the release of LH rapidly and in increasing This is in the early secretory phase, shortly after ovulation. There will be vacuoles with subnuclear intracytoplasmic granules appear in the glandular cell. This vacuoles progress to the apex of the cells and the content are released into the endometrial cavity.